Good morning everyone. Today, we are going to delve into the intriguing world of cells, understanding their structure and function, and exploring the biological principles that control their behavior. Come along with us as we gain a better understanding of the mechanisms of cells. We'll be discussing the core concepts of cell theory and its structure. We will start by defining what a cell is and examining the cell cycle. Next, we'll look into the meiotic phase, followed by the phases of cellular respiration and the glycolysis pathway. By the end of our exploration, you should have a greater comprehension of the structure and theory of cells. Biology is the study of complicated things that have the appearance of having been designed with a purpose, a famously stated fact by Richard Dawkins. Matthias Schleiden and Theodor Schwann proposed the cell theory, which radically changed our understanding of living organisms and established that cells are the basic components of life. Proposed in the mid-19th century by Matthias Schleiden and Theodor Schwann, cell theory is a cornerstone of biology and has revolutionized our perception of living organisms by asserting that cells are the fundamental units of life. It is based on the idea that all living things are composed of cells and these cells are the basic units of structure and function in every living organism. This has allowed us to gain a better understanding of cellular structures and functions, leading to other important concepts such as metabolism, reproduction, and growth in biology. All living organisms are composed of cells, which are the basic building blocks of life. Cells, the smallest unit of life, are not created from nothing. They are made up of other cells, and all contain genetic material which allows them to reproduce and form new cells. Cell structure determines their function, with some being specialized and others more generalized. Cells may come in a variety of shapes and sizes, and are organized into tissues, organs, and systems in multicellular organisms. Cells are the basic building block of all living organisms and their structure and function is integral to understanding life. An outer membrane acts as a barrier and controls which materials enter and leave the cell. Inside cells are special organelles, allowing for the production of energy, reproduction and storage of proteins and genetic material. These components work in unison for the cell to survive. Prokaryotic cells, like bacteria and archaea, have a simpler and more efficient structure and design. These cells do not have a nucleus or organelles, and contain only a single circular chromosome. To make up for this, they have cell walls for structure and protection, as well as ribosomes for protein synthesis. During the cell cycle, the sequence of growth and division of a cell begins with the gap 1 phase, a period of cellular activity and preparation for DNA replication. During this phase, cells grow in size, synthesize proteins, and carry out their specialized functions. The next phase is the gap 2 phase, during which cells continue to grow and prepare for division. Necessary proteins, organelles, and other components required for cell division are synthesized. Chromatin, the loosely packed form of DNA, condenses into distinct chromosomes at prophase. At prometaphase, the nuclear envelope disintegrates and a spindle apparatus, composed of microtubules, forms to facilitate the separation of chromosomes. During metaphase, the chromosomes are aligned along the cell's middle, or the metaphase plate. Meiotic phase, the process of separating chromosomes during cell division, is complete at this stage. Prokaryotic cells are the most primitive and most common form of life on Earth. Lacking a nucleus or organelles, prokaryotes are single-celled organisms that consist of a cytoplasmic membrane and cell wall. Inside the membrane are small circular pieces of DNA called plasmids and other proteins and molecules. Prokaryotes rely on osmosis to regulate pressure and obtain nutrients from the environment. Their size allows them to be highly efficient in harvesting energy from the environment, and they can reproduce quickly. In prokaryotes' reproduction, binary fission is an asexual process that involves two steps. First is the replication of the single DNA molecule attaching to the membrane and then the growth of the cell membrane, doubling the size of the cell. Afterward, the cell wall is formed producing two daughter cells. The basic cell theory provides foundational knowledge about the structure and function of prokaryotic organisms. Recombination plays a critical role in genetic variation in these organisms, allowing them to adapt and survive in different environments. This process involves combining genetic elements from two or more DNA molecules to form a new one. This new genetic information can create new organisms with unique characteristics. A better understanding of recombination and its implications can help us gain insights into the fundamentals of life and the evolution of cells. Cellular respiration is a process inside cells in which nutrients are transformed into energy. Without cellular respiration, cells would not be able to exist. During the process, oxygen is consumed and carbon dioxide and water are produced as byproducts. Cellular respiration is significant for energy production, allowing us to do the necessary activities for survival. Glycolysis is the process by which glucose is broken down into pyruvate, releasing energy for the cell, while the citric acid cycle is the metabolic pathway by which cells generate energy from oxygen and other molecules. Both of these processes are essential for understanding how cells function, as they form the basis for cellular metabolism and respiration. Glycolysis is a pathway of enzymes that breaks down glucose molecules into two pyruvate molecules, providing energy to the cells in the form of ATP molecules. In addition to energy production, glycolysis is also essential for synthesizing new molecules, as without it, cells would not be able to create new molecules that are necessary for growth and survival. Understanding the basic cell theory and structure is essential to gain insight into the complexities of diabetes and genetic diseases. Cell theory explains that cells are the fundamental unit of life, and understanding this can help us comprehend how these conditions come to be and evolve. With a deeper knowledge about cell theory and structure, researchers are able to make advancements in the study of diabetes and genetic diseases, and ultimately help lead to improved treatments. The citric acid cycle, also known as the TCA or Krebs cycle, is a metabolic process that involves the breakdown of carbohydrates, proteins and fats. During this process, the molecules of these macronutrients are broken down and energy is produced in the form of ATP. This energy is then used by our cells to power their processes, essential for sustaining life. The Krebs cycle is a series of biochemical reactions that happen inside a cell's mitochondria. It is a crucial process for the production of energy necessary for cellular work. Following a loop pattern, each of the 8 steps of the cycle produces a molecule that is used to initiate the next step. To help recall the cycle, an easy acronym has been devised. Cannot pass through an open gate. Each letter of this acronym corresponds to one of the 8 steps of the Krebs cycle. We will discuss the basic cell theory and structure, exploring the protein complex series from food to energy, and the electron transport chain. Both of these processes are essential for complex living organisms, and to understand cell biology, it is important to understand them. The protein complex series takes in food molecules and converts them into energy, while the electron transport chain takes this energy and packages it into ATP, which in turn powers the cell and its molecules. We hope this discussion has helped in understanding these complex processes in further depth. Thank you for your attention.